Hi guys, it's 7, so we're gonna get started. Did everyone sign in? I'll give you guys a second to do that. Okay. Again, here are the same announcements. Office hours are tomorrow, 10 to 11 in row. I think it's room 217, if you don't know where the Academic Achievement Center is. There's my email. Feel free to email me anything at all. Also, a reminder, I'm involved with like research and stuff, so like if you have questions about that, I can also help you. And then my notes. I think my notes are up for endocrine stuff. If they're not, like send me an email and I'll put them up. Here is a feedback survey. Some of you guys before break did it. If you weren't here before break, if you could do this really quickly. It's just a few questions to help me understand like what's helpful for you guys, what I can improve on. I got a lot of feedback that you guys would want more like practice questions, so I did some more practice questions in this one. But yeah, it's really helpful to look at. So I'll give you guys like a few seconds to do that. Okay, since a lot of you are here last time, I'll move on. Okay, this week we're talking about the endocrine system. So we've talked a lot about nervous system control with neurotransmitters and neurons. So we'll just move on to the next slide, which compares the nervous system to the endocrine system. Nervous system is electrical signaling, while the endocrine system is chemical signaling. So back when we talked about the nervous system, we used neurotransmitters to go just from neuron to neuron, which is just like one cell to another cell, which is very short, small, short signaling. But when it sums all together, you can go far distances. But in one unit, I'd say it's short signaling. Endocrine signaling, on the other hand, is systemic signaling. So you're getting a hormone released from a certain tissue in the body, and then that hormone is going to enter your bloodstream and travel a far distance to a bunch of different receptors on tissues in your body. So that's also known as global signaling or systemic signaling. And another thing to note is that there's a longer recovery period when you have an endocrine system. So a hormonal signal is going to last longer than a nervous system signal. So there's another type of classification that's in between nervous and endocrine signaling, and this is known as neurohormones. So this is like an exception between the two systems. It's when a neuron directly secretes something into the bloodstream that then initiates a response in a target tissue instead of going neuron, neurotransmitter to next neuron. Do you guys have any questions on the difference between the two systems so far? Okay, cool. So here's just like a picture of the two systems. Like we were talking about before, with the nervous system, you have the brain and spinal cord, and then all the peripheral nerves. And then the endocrine system consists of the hypothalamus, pineal gland, pituitary gland in the brain, and then you have the thyroid gland, the thymus, pancreas, adrenal glands on top of your kidneys, ovaries, and testes. This is a picture of the neurohormone signaling. So it's just the neuron releasing some sort of signal or neurohormone into the bloodstream that then goes to a target cell. in that target cell can be like any tissue throughout the body anywhere. So here's some more information about endocrine tissue. It's a type of epithelial tissue, and it's organized in glands. So glands produce and release hormones into nearby blood vessels. So the endocrine tissue is kind of like organized into little kind of sacs that produce hormones, and then the hormones are released into the blood. And the endocrine tissue is decentralized, which means that it's all throughout your body. Like we saw in this picture, like you have some endocrine tissue in the brain, your like throat area, chest, all the way down into your abdomen, whereas the nervous system is more centralized into your brain and spinal cord. And this is possible because it allows secretions into the bloodstream. So then at the bottom, that's kind of small, but you have the slides. I think you go over more of this in lab, but those are just like microscope pictures of the different types of endocrine tissue. So there's thyroid, adrenal, thymus, ovary, pancreas, and testes tissue there. And you can see like the organization of the glands. So here are some processes under hormonal control, so some functions of the endocrine system. Metabolism controls breakdown of fats and the release of glucose. So two hormones that are important in metabolism are glucagon and insulin. So for example, like if you have high glucose, you're going to release insulin. And then insulin has like a very systemic effect on the whole body, helping your body like store glucose. So it has an effect on multiple tissues at one time because of a certain state that your body is in. Vascular function can increase or decrease heart rate. So for this one, a hormone that would be responsible for that is like epinephrine. When you're like exercising, your heart rate increases, your breathing increases. All of that is regulated by like in one hormone that has a bunch of different receptors all throughout the body that have a different function. Reproduction is also under hormonal control. You can think like estrogen, testosterone. Cell growth and proliferation. This is regulated by like growth hormones that then have different receptors in the body. And then the hormone will bind and induce some sort of response that like regulates growth. Cell differentiation, that's when like stem cells become specialized between like a muscle cell or like an epithelial cell. So hormones play a role in that process and behavior. This slide's kind of just emphasizing that there are like a lot of different hormones in the body, and they vary in where they're produced. So we just talked about a bunch of different endocrine tissues. There are different hormones produced in different tissues. They all have different structures, and structure determines function, so they all have different functions. Here's a breakdown of the three main hormone classifications. I just put this on one slide so you can like see them all at once. So starting off with peptide hormones, these are genetically encoded. You can think like proteins. That's how I think of like peptide hormones, proteins. Genetically encoded. They're produced inside the body from genes being transcribed and translated. Some examples of peptide hormones are releasing hormone, glucagon, and insulin. And all peptide hormones are water soluble, which means that they do not need a carrier protein when they're transported through the blood. They can just dissolve right into the blood plasma and go where they need to go. and that allows them to have a lot shorter half-life. They travel through the body pretty quickly without the use of a carrier protein, so they're a little bit faster than lipid-soluble hormones. Steroid hormones are based off of a cholesterol backbone, and then here's just a quick definition of cholesterol. A cholesterol just contains 4 hydrocarbon rings altogether. So that makes it very lipophilic, which means that it can dissolve through membranes, but it cannot dissolve in water. So it needs a carrier protein when it's being transported through the blood. It has an R group or a side chain, which allows for variation. And then there are 2 types: sex steroids, which include estrogen, progesterone, and testosterone, and corticosteroids, which are from the cortical region of the adrenal gland. And then these are cortisol and aldosterone. And then the last classification is amine hormones. And then these are derived from one of 2 amino acids. The first one is tyrosine. Amine hormones from tyrosine are epinephrine and norepinephrine. It also makes the neurotransmitter dopamine and thyroid hormones. The other amino acid is tryptophan. And tryptophan is used by the pineal gland and the epithalamus to produce melatonin, which is the hormone. And melatonin is important for regulating sleep-wake cycles. And melatonin also gets further processed into serotonin, which is another hormone. And most amine hormones are water-soluble, which means that they can travel through the blood without a carrier protein, but thyroid hormones are lipid-soluble. So that's just a little distinction to make. Any questions on the classifications? Okay. So this slide just further breaks down the difference between water versus lipid solubility. So again, water-soluble proteins are peptide in most amine hormones except for the thyroid hormone. And these do not need a carrier protein in the blood. They can just dissolve right into the plasma. So they have a much shorter half-life. They travel through the body and they finish their process a lot faster. Lipid-soluble hormones are lipophilic. These are the steroid hormones, and then the thyroid amine hormone. They must be bound to the carrier protein when they're transported through the blood. And they have a lot longer half-life in the body. So it takes a longer time for them to be removed from the body once they're produced and released into the blood. They're going to have a longer lasting effect on the body. Another important distinction that I didn't put here is that once the hormone reaches its target tissue, water-soluble hormones need to bind to a receptor, whereas lipid-soluble hormones can just pass right through the cell membrane of the target cell because they're lipid-soluble and the cell membrane is mostly made of lipids, which we get more into later, but I just wanted to add that on here. Here are the ways that peptide and steroid hormones are made. So peptide hormones are pretty easy to make. They're synthesized as pre-prohormones in the endoplasmic reticulum. And proteolytic enzymes break some of the peptide bonds. These are inactive, and they're processed into prohormones, which are even smaller because some of the peptide bonds were broken. And then they're further cleaved in the Golgi apparatus into the active peptide hormone. So basically they just start out as like a bigger peptide, and then they just get cleaved down in the Golgi apparatus, or in the endoplasmic reticulum, get more cleaved down in the Golgi apparatus, and then they're in the active peptide hormone form. And it's the smallest one because it kept getting break it down. Steroid hormones, this is a more complicated synthesis. So you don't really have to know the details very much, but cellular enzymes modify cholesterol backbones to produce different hormones by altering their R groups. And a very small difference in structure can make a very different hormone and have a very different effect on the body. So the example I think is given in lecture I put there is testosterone is a secondary sex characteristic hormone, and then that enzyme can make it turn into a hormone that like causes baldness by like a small adjustment. So this is just basic hormone action. So hormones are known as the first messengers. They're produced by endocrine tissue somewhere in the body, released into the blood, and then they travel through the blood to some sort of receptor on some sort of target tissue. And the receptor is specific to that hormone. So your body can upregulate or downregulate receptors based on how it wants to respond to the hormone. So receptor upregulation will be putting more receptors on the tissue, increasing sensitivity to the hormone, whereas you can downregulate the receptor, and then that would decrease sensitivity to the hormone. So if you put more receptors on a tissue for a specific hormone, then that's going to increase that hormone's response. Or you can decrease receptors and decrease that hormone's response. And then the last bullet is just kind of saying that the type of receptor and type of hormone matters. Like if you increase insulin receptors, then that's not going to have any effects on like glucagon activity. You'd have to increase the specific receptor for the specific hormone to increase that hormone's response. And then this picture just shows like the endocrine tissue releases the hormone into the blood, which travels to the target cell, which has the receptor on it. This is what I was talking about before, the difference between hormone transport into the target cell based off of the hormone solubility. So again, water-soluble hormones, peptide, and most amine hormones, they do not need a carrier protein in the blood, but once they reach the cell, they need a receptor because they're water-soluble and they can't travel through the membrane. So if you remember, like, the cell membrane is like this, like the hydrophilic head and hydrophobic tails. So the hydrophobic region is a lot bigger in the cell membrane. So the water-soluble hormones like can't pass through this like huge region of hydrophobic, but the lipid-soluble hormones can pass through this big hydrophobic region. So the lipid ones don't need a carrier protein. They can just diffuse right through. But water-soluble hormones are going to have a receptor that they're going to bind to, which is going to induce some sort of like cellular modification or change on the inside of the cell. So they don't need to actually pass inside the cell. And so this system where the water-soluble hormone binds and then it induces structural changes inside the cell is called the secondary messenger system, where the signal is transduced into the cell using like enzymes. So for lipophilic hormones that pass right through the membrane, another point about those is a popular function is gene transcription. So those can pass right into the cell, and they have intracellular receptors. So instead of receptors being on the outside, they have some sort of receptor in, like, this is the cytoplasm, or on the nucleus itself. And they regulate gene transcription often, which like makes sense because of that. An example is estrogen, which is a lipophilic steroid hormone, and the receptor is on the nucleus, which turns transcription on or off for a lot of different things, including cell growth and proliferation. So here's the same thing I just said, but in a diagram form. I like keep saying it over and over again, but like the water-soluble ones travel through the blood, go to the receptor, and then the lipid ones need a carrier protein, go right into the cell. So there are two types of receptors for water-soluble hormones on the extracellular side of the cell, known as G protein-coupled receptors, or GPCRs, or receptor, and then another one is receptor tyrosine kinases, which is RTKs. The first one is GPCR. So this is just the general structure of a GPCR. This is the most common receptor on the plasma membrane that responds to water-soluble hormones. So there are 3 subunits. And so the first one is alpha, and then there are 3 different forms of alpha. You have G alpha S, G alpha I, and G alpha Q. So yes, G alpha S activates adenyl cyclase. So when a hormone binds, what happens is there are these 3 subunits, and like one of them will like release and activate some sort of enzyme in the cell that initiates some sort of response. And it'll usually activate a kinase, which phosphorylates something, which activates it. So if we look at G alpha S specifically, this one, when it dissociates, it'll go activate adenyl cyclase, which is just another protein in the cell membrane. So just to give you guys context, the hormone will bind here. The G protein receptor is all the way through the membrane. So then the alpha subunits, the 3 of them, are like inside the cell. When the hormone binds, G alpha might dissociate and then bind to adenyl cyclase, which is just another like membrane protein. Adenyl cyclase will be triggered to make CAMP from ATP, and CAMP is known as a secondary messenger. So once adenyl cyclase is activated, it produces CAMP. And then CAMP can go to activate protein kinase A, and then protein kinase A is a kinase that will then go phosphorylate something and turn it on or make it active. And then two other subunits of the GPCR are beta and gamma. So here are the components, like more basic. You have the hormone or neurotransmitter that binds to the extracellular side of the extracellular receptor. Then you have the transmembrane protein, which is the part of the GPCR that just like spans the membrane, and it like transmits the signal to the three subunits on the inside. And then the enzymes inside the cytosol are then activated, depending on what hormone activates the G protein. So here's an example. Epinephrine or norepinephrine binds to the extracellular receptor. It causes the transmembrane to change conformation, which allows the alpha subunit to dissociate. And then that can go activate something like adenyl cyclase, which then might turn ATP into CAMP, which then might activate a kinase. Another thing to note is that phosphorylation, or just like adding a phosphate to something to turn it on, can happen on one of three amino acid residues: serine, threonine, or tyrosine. Yeah.
So phosphorylation only happens on those amino acids?
Yes. And then here's a diagram. You got the three subunits. You can see that alpha dissociates when when a hormone or like ligand binds, which allows the alpha subunit to go activate something and produce a cellular response. And then here's the same picture that I think is used in lecture, just to tie it in. Oh, so something else that happens is when the alpha subunit is released, it exchanges GDP for GTP, and it's just like activated. The transmembrane domain changes conformation, alpha is released, alpha exchanges GDP for GTP, and then goes and activates something like adenyl cyclase. So the other water-soluble receptor is the RTK. This one's a lot simpler. There are not as many components, and they're all in one protein. So you have the first messenger, which is, again, just the water-soluble hormone. And then you have another transmembrane receptor, which is coupled to tyrosine kinase. So what happens with this one is you have a water-soluble hormone that binds to the extracellular receptor. You kind of have two domains of an RTK, and then the binding of it kind of makes the two domains come together, and then that induces cross-phosphorylation of the RTK, which activates it. And then that's basically it. So then it's activated and can phosphorylate something. So basically, binding of a hormone to the RTK causes the RTK to phosphorylate and activate itself. And then the RTK can activate something else inside the cell. And RTKs are often involved in cell growth and proliferation. So here's a diagram of what I tried to just explain. You have a hormone, which is typically a growth factor, because these ones do a lot of cell growth and proliferation. So when the hormone binds to the extracellular receptor, it causes the RTK, you can see it's made of like two subunits, to come together, and then they activate and phosphorylate themselves, and then it's activated. And then here's another diagram of the same thing, if this one's better for you to look at. I like how this one also adds that pink protein at the end. So you can see the activated RTK, and then you see the pink inactive protein comes in contact with the RTK, and then it's activated. Which leads to a cellular response. So an example of an RTK is chronic myeloid leukemia. So in chronic myeloid leukemia, the kinase or the receptor tyrosine kinase is always active, even without a water-soluble hormone turning it on. So you can see how that would be problematic, because you have an RTK that regulates growth and it's always on, so you have uncontrolled growth and proliferation, which leads to cancer. So there's a drug called Gleevec or imatinib, I don't know if I'm saying that right, and it blocks the active site of the RTK, which prevents the kinase from phosphorylating substrate proteins. So where that pink protein would have bound, this drug blocks. So since RTKs tend to mutate, this drug can't really be used on its own, and it needs to be used in combination with other drugs to protect against the RTK mutating and overcoming the effects of the drug. Okay, I think I have a bunch of practice questions. So first one is, what is a common characteristic of all steroid hormones? Yeah. E. Okay. So receptors are inside the cell and they are soluble in the membrane. Yes, that is correct. So if you look back here, steroid hormones are lipid soluble, travel right through the membrane, have receptors inside the cell, and they're soluble in the membrane. Same question, but in reverse. What is a common characteristic of all peptide hormones? Yeah. So we got receptors are on the extracellular side, and they're impermeable to the membrane. They can't cross through because they're water soluble. Which hormones are water soluble?
glucagon insulin melatonin?
Yes. So a way to like remember which ones are which, like the lipid soluble ones are kind of easy to remember just because you kind of know them, like estrogen, progesterone. I have estrogen on there twice. And then thyroid hormone is the exception of the amine hormones. What two amino acids are the amine hormones derived from? Yeah. Yes. Perfect. True or false? Water-soluble hormones have a longer half-life in the body than steroid hormones.
False.
Yeah. So water-soluble hormones don't need a carrier protein. They go right through the blood pretty fast, faster response. Last shorter time. True or false? Upregulation of a hormone's receptor increases sensitivity to that hormone. Yes. So if you downregulate a hormone's receptor, you're going to decrease sensitivity. Hormonal signaling is blank, while neuronal signaling is blank. That works. That's not what I was thinking. I was going for like the scale. Yeah.
Hormonal signaling is more like global,
Mhm.
and neuronal is more like centralized.
Yes. Both of them are right, but I said systemic and localized. True or false? Steroid hormones need a carrier protein when being transported through the blood. Yeah. True. True or false? All amine hormones are water soluble. False. Thyroid hormone is lipid soluble. What are two types of water soluble receptors on target tissues, and what do they do and why? So does anyone know what the two types of receptors are? Yeah.
GPCRs and RPK.
Yes. And does anyone want to try what they do and why they do it? If not, it's okay. Okay. Oh yeah, go for it.
They cause an intracellular response
Mhm.
to phosphorylation.
Yeah. Yeah. So these basically they're transmembrane receptors that induce an intracellular response without the hormone needing to travel through the membrane because water soluble proteins can't travel through the membrane. So this is just how like the cell can know that that hormone is there by inducing a change in the intracellular domain of the receptor, and then it triggers some sort of cellular response without the hormone actually physically having to enter the cell. And then that's all I have. If you have any questions, feel free to come up and ask or email me. Yeah.